let's wrap up Chapter 6 with Lecture Video C. We'll cover slides 25 through slide 30. So before we move on, let's kind of wrap up and kind of recap what it is that we've been talking about so far. We learned that we can grow microbes in a pure culture in lab where it's all one single species, genetically identical cells. We said that out in the natural environment, typically we don't find a single species living kind of all on their own. We're going to have mixed species all living together. We looked at how environmental conditions and oxygen availability can influence our microbial growth. We're now going to look at an instance where we can have multiple individual prokaryotic cells working together for the benefit of the group as a whole. We're going to talk about biofilms. So a biofilm is a mass of bacteria that are stuck to one another and they are anchored to a surface. Now, it is possible that a biofilm can consist of a single species of bacteria, but more commonly, these guys exist as mixed communities where there are multiple species of bacteria living together. Sometimes they will be competing, but in the case of a biofilm, they're actually going to all be working together for their collective good. So the surface that a biofilm can form on is really diverse. Like there's not a lot of rules that we set forth for where we can find a biofilm. It's possible for a biofilm to form on the surface of living tissues. One example of a time where we can have a biofilm that consists primarily of a single species is a bacteria called Pseudomonas aeruginosa. This is a particular bacteria that we use in labs. Sometimes we will grow it up. If you've ever grown it on a petri dish, it has kind of a blue-green tint to the bacteria, and so it's one of those that we can spot really quickly when we're using it in lab. But this particular bacterial species can be inhaled and it can begin to grow on the interior part of those lungs. It can, because of this biofilm formation, it can be really, really difficult to treat. Now, there are times where we can have other organic substances that a biofilm can form on. We can have inorganic substances as well. The classic example of a biofilm is depicted here in this picture. It's totally gross, but biofilms can form on your teeth. In fact, everybody has biofilms that will form on their teeth by the different microbes that inhabit your mouth. So no matter how much you brush or how great your oral hygiene is, everybody is going to have plaque that forms on the surface of the teeth. So in this particular image, what they've done is they've had the person chew up a tablet that stains the plaque, that stains that biofilm. Sometimes we'll do this with children to show them where they need to brush a little better. Quality brushing, making sure we get up into all the little nooks and crannies, can certainly help reduce the amount of biofilm, but this is a really tough structure and it's hard to get rid of with just brushing. So one of the reasons that you should go and visit a dental hygienist every 6 months or 1 year, depending on your insurance, is so that they can go in and use a metal scraper and actually go in and mechanically remove that really tough biofilm. Let's look at how a biofilm actually forms. So this picture down at the bottom depicts the different steps that we're going to take to form a biofilm, and then we'll look at why we might want to disassemble our community of microbes. So we begin, get my mouse here, we begin by having a few cells settle on our surface. Now, in this particular instance, they show flagellated cells, meaning that they have a flagella. They don't necessarily have to have that, but they're going to attach themselves to a surface. This is just a few cells. They're going to produce additional secretions in their area to help recruit other individual cells. they're going to slowly begin dividing. They're in an area where there are nutrients available, and so they're going to undergo binary fission. As part of their normal processes, they are going to be producing chemical signals. So up till now, we've really kind of led you to believe that microbes live a really solitary life as single-celled organisms. And that's not actually the case. They have the ability to register the presence of other cells by using these secreted chemicals. So each cell makes a certain amount of these communicating chemicals, and as there are more cells, that concentration of those signaling chemicals increases and increases. The more cells you have, the more of that communicating molecule you're going to have. We refer to what is called quorum sensing, where the cells are not just making those signaling molecules, but they are registering the concentration of those signaling molecules in their immediate area. When the chemical signals reach that specific concentration, we say that a quorum has been reached. Oftentimes we hear this in terms of like politics. It's kind of the same idea. So we've reached like this critical threshold, and it tells the cells, hey, there's plenty of bacterial cells here. Maybe they're of the same species, maybe they're of different species. But once we have enough cells there, they can collectively work together. So reaching that quorum allows these microcolonies to begin undergoing genetic changes. One of those things that is going to change is the production of a substance called exopolysaccharides. These are carbohydrates, and they are produced externally to the cell. So that's how we break down that name, exopolysaccharides. There's a bunch of different types. This creates a really thick, kind of goopy, I don't know, kind of a slimy texture, consistency to it. So this matrix of carbohydrate is now surrounding all of the cells. It comes through all of the cells. This exopolysaccharide acts to help trap various substances. We will see that those substances can be moved through that exopolysaccharide. So if we need to get nutrients to the cells that are at the most interior, we can do that. This exopolysaccharide is going to, oh goodness, my light's turned off. Ah, there we go. I'm in my office recording today. Those exopolysaccharides are going to limit antibiotic access to those cells. So it acts as almost kind of like a blanket on that most external part. And it's going to help to protect the cells from antibiotic compounds. When we talk about Pseudomonas aeruginosa, that's one of the reasons that this particular bacterial infection is difficult to treat, because it's hard to get the antibiotics directly into the cells to act on those cells. So it's good for the bacteria that are living in this community, but if you are a person who's dealing with the infection, it can be a long treatment process. So this biofilm is going to continue to mature. It's going to continue to have cells divide as long as those nutrients are ample. If those nutrients become depleted or conditions change and that environment is no longer favorable, then the cells will begin to stop making that exopolysaccharide, and that kind of blanket-like structure is going to begin to degrade, and it will allow the cells that are still living to be released, so that they might stumble upon some other environment and set up a new biofilm elsewhere. So biofilms are really interesting. They can make treatment of some bacterial infections particularly difficult. All right, so we have talked a little bit about the formation of endospores. We said that spores are dormant structures that certain genera of cells can make. So the two genera that can produce endospores are the Bacillus genus and the Clostridium genus. These are both gram-positive, so they're going to have that nice thick cell wall of peptidoglycan. This process of making endospores is going to occur when conditions become harsh. I like to think of endospore production as kind of like backing up your hard drive. So if your computer is a living vegetative spore, it's good, it's doing its thing, but it starts to act a little glitchy and you're like, oh my gosh, I have all this information on the computer and I don't want to lose it. Your vegetative cells of these particular genera are going, oh my gosh, I need to back up my genetic information. So much like you might back up your computer to a hard drive so that it can be installed later on a different computer, you can still save that information. That's kind of the idea behind our endospores. We're going to take the most critical information and we're going to save it. So up till now, we've talked about this vegetative growth. We said that our DNA polymerase is going to replicate our chromosomes beginning at that origin of replication. We're going to separate our two daughter cells, binary fission. Each of those becomes a new parent cell, undergoes binary fission, time and time and time again. So typically, this is what these two species of these two genera are going to do. But when conditions become harsh and they need to back up their hard drive, they need to form an endospore. They undergo a process called sporulation. They will only do this if conditions become harsh. So what happens is that we copy that DNA chromosome, and we are going to have what is called offset septation. Remember that a septum is that dividing line. Well, here we're going to have it offset. It's not going to be right in the middle. We're going to have one whole chromosome that's going to go to one side. The other chromosome is going to go to the other. We are going to complete that septum. The smaller of the two is going to be called the forespore. Eventually, this is going to go on to become the endospore, or just the spore. The mother cell is the larger portion. We'll have the mother cell extend and engulf that forespore. What that does is it creates a forespore that has two membranes around it. So look, we have two membranes here. That space in between is called the cortex, and we're going to form a thick layer of peptidoglycan in that open space between the two membranes. We're going to have a spore coat that's going to form on the outside of that. We are going to have dipicolinic acid that is produced. And eventually, we're going to see the total degradation of the remnants of that mother cell. And all we're going to have left is this spore. This is like our external hard drive. It is incredibly resistant to drying out. We call that desiccation. So it won't desiccate. It won't dry out. It can withstand incredibly high temperatures for long periods of time. It doesn't need a nutrient source. Spores are very long-lived, tough structures. And they will stay in that spore form until conditions become favorable again. And when that happens, we'll go through a process of switching back to that vegetative state and actively dividing. If conditions become harsh, we go through that sporulation again. So I've got an endospore formation animation that I'd like to show you. The one on anthrax is available if you're curious about this type of infection, but we're not going to watch that one right now.
Spore-forming bacteria grow as vegetative cells and divide by binary fission when nutrients are available and environmental conditions are not adverse. When nutrients are depleted or conditions become adverse, spore formation is initiated. The DNA condenses and aligns itself in the center of the cell. The The vegetative cell is now referred to as the mother cell. Next, the DNA divides into two complete copies, and the mother cell membrane invaginates to form the developing forespore. The mother cell membrane continues to grow and engulfs the developing spore. The developing spore is now surrounded by two membrane layers. Next, peptidoglycan is laid down between the two membranes of the developing spore to form the cortex. Dipicolenic acid is formed inside the developing spore, and calcium enters from the outside. As calcium enters the spore, water is removed. A protein coat forms exterior to the cortex, and the spore becomes mature. Some spores form an additional layer called the exosporium. A mature spore is resistant to environmental conditions. Finally, lytic enzymes destroy the mother cell, and the mature spore is released.
All right, so endospores are incredibly significant in the fact that they allow these types of bacteria to become infective at a much later date. And there are some very serious diseases that a couple of these species cause. So we've already talked about Bacillus anthracis, which is the causative agent of anthrax, but Clostridium botulinum, the causative agent of botulism. And then we have Clostridium tetani, which is the causative agent of tetanus. Those diseases are incredibly dangerous, potentially. And so when we look at the way we can pass these bacteria from one location to another, endospores mean that they can be very readily transmitted from one area to the other, even though we might not have good conditions for growing bacteria. At that time, they can still be, those spores can still be transmitted. And as soon as they encounter those good conditions, those favorable conditions, they'll go back to that vegetative form, potentially causing major, major issues within the human body. We have several notes on places that you need to read throughout Chapter 6. I've tried my very best to be specific so that you are reading only what you need to read. Your study assignment questions are just objective review questions this time. There are several of them, but there are no short answer review questions. Please let me know if you have any questions.